This
Allison,
conference will now be recorded.
and I'm one of the co-chairs of the HESI E-STAR committee along with Cam Johnson. The E-STAR committee is the Emerging Systems Toxicology for Assessment of Risk committee. And for the last couple of years, we've had a webinar series approximately quarterly on topics that we think would be of broad interest to the committee and would have impact on the committee's activities. Just a little bit of housekeeping items. This webinar will be recorded. During the talk, please feel free to type questions in the chat box. We'll wait till the end of the presentation to address them, but you can type them in at any time. And when we reach the end of the presentation, if you would prefer to ask your question verbally, feel free to raise your hand and you can unmute at that time and go ahead and ask your question. All right, and then without further ado, I'm pleased to introduce our speaker today, who is Richard Currie. Richard is a Syngenta Fellow from the Syngenta Gellets Hill International Research Centre in the UK. And there he leads the human safety science support for the crop protection chemistry research portfolio. He serves as an internal consultant on investigative and mechanistic toxicology and is an entrepreneur for predictive and computational approaches for product safety assessment and chemical design. Dr. Currie previously has led activities on mechanisms of non-genotoxic carcinogens using a toxicogenomics approach and has employed the use of epigenetics in toxicology. His interests extend to development and application of new approaches to toxicity prediction, including structure-activity relationships, mechanisms of toxicity, including mode of action and AOPs, and systems models for ADME, toxicology, ecotox, and environmental fate. So Dr. Currie, we're delighted that you're with us today, and when you're ready, you can take it away.
Thank you, Alison. Hopefully everyone can see the screen and see my laser pointer. I'm going to take probably about 40 minutes or so of talking, and then we have plenty of time for a conversation afterwards, I think. So please feel free to ask loads of questions towards the end. I'm mostly going to be talking about a project that's being a working group in the HESI DART committee on the DARTable genome. So we are elaborating and quantifying the DARTable genome to predict teratogenic doses is the grand title. We haven't quite got there yet, but hopefully I will convince you that this is possible and that we will have I think, potentially a fruitful collaboration between the two working groups as part of that in the future. So I'm going to start with the DARTable genome hypothesis. I'm going to briefly talk through the journey that got us to the starting point of the HESI DARTable genome working group, go through that group's current activities, and then finish up with some things about what we plan to do next and where we might fruitfully collaborate. So what is the DARTable genome hypothesis? It's effectively the subset of the genome that, if it was perturbed, results in a developmental or reproductive effect, by analogy to the druggable genome. But effectively, it's the subset of relevance for certain toxicities. When you think about this in a bit more detail, actually, there's a couple of different possibilities about what when perturbed might mean. It could be that the gene products, so the proteins, are direct targets of an exogenous toxicant. So these could be things that identify molecular initiating events, or it could be that the gene products or have their abundance has changed in response to a perturbation, be it chemical or genetic or disease associated. And these effectively are the biomarkers that might be used to identify the activation of a pathway that results in developmental toxicity. So conceptually then, the DARTable genome is the comprehensive set of possible molecular initiating events and the transcriptal proteomic biomarkers for those events. And you might use different approaches to identify both of those things. And I hope by the end of the talk, I'll have provided some first illustrations of how we might go about doing that. So, our journey. For us in Syngenta, this started with an ACCA's herbicide project. This is an enzyme that's important for fatty acid biosynthesis. And the one thing that we knew at the time we started this project was we didn't really have a problem with teratogenicity, at least we thought we didn't. No registered ACCAs has had this problem. We hadn't seen this before in historic molecules as far as we knew at the time. And we had a number of candidates that were really potentially useful for control of weed species in cereals, rice and soybean. So some very important commercial crops. Unfortunately, that wasn't to be, because we identified a very clear signal of body wall closure defects that were seen in a number of compounds. It's actually many more than just the 3 we detected here. We saw this on phalloceal with hind limb deficit in a number of molecules in both rats and rabbits. And all the substances we tested, they weren't mutagens, they were high purity. We screened in Panlab screens to see if they were promiscuous to other targets. We weren't seeing anything. So we were thinking, okay, this is an interesting one. Where do we go with this? So could it be the ACCA's mode of action, even though historically we haven't seen that? So, well, the answer is possibly. Yes, it could be, because we had very low potency for the human enzyme. Even though we had nanomolar potency against the plants, it was still wonderfully selective, but not completely specific. So we did get some possibility of ACCA's inhibition. So could it be that ACCA's inhibition is important for development? Well, possibly, because we know that ACC1 knockout mice are embryonically lethal. So this is, as you might imagine, a very important biochemical process that is essential for the development of a mammal. So we looked further into the literature, and actually there are a number of pharmaceutical ACCA's inhibitors that have been developed for a variety of indications. But ACCA's inhibition produces fatty acids, which are important in hedgehog and Wnt signaling. It's been shown to, inhibition of that has been shown to affect endothelial cell migration and invasive podia formation in cells. In C. elegans, actually, there's the first cell division in the C. elegans development is asymmetric. You knock out the C. elegans ACCAs and it's a symmetric division, and the nematode doesn't develop properly. And that can be rescued by adding fatty acid back, again showing that polarization in that first division is important. And you get similar effects with lipid perturbations in C. elegans intestinal cells. We did a lot of bioinformatic work just mapping the phenotypes to show that there were actually 66 other alleles that generate this omphalocele phenotype when knocked out in mice. And then when you look at what are the upstream and downstream regulatory consequences of those genes, you get very similar thing. You get the cytic hedgehog pathway, Wnt signaling, and you also get this planar polarity cell specification. So there's a couple of biologically plausible mechanisms through which inhibition of ACCAs could give rise to developmental defects. And then we looked at the exposure of these herbicides. So these are, down here, are typical herbicides. And these are the AUCs in rats that you see on a single dosing. This is what we were getting with this new chemistry class. So effectively, we thought, oh dear, it's exposure driven despite the low potency. And that's what we explored in these pharmacokinetic studies, where this is in vivo PK out to just a 24-hour single dose time course. And you can see exposure on the x-axis, so it's a blood time profile. And for each of these different compounds, I've highlighted where the enzyme Ki value is. And when you see a cross or a tick, it means that in the DevTox study, at that dose, there was a teratogenic effect. And if there's a tick at that dose, there wasn't a teratogenic effect. So what you can see is we've got this interesting correlation here between doses above the threshold, Ki value, where you see teratogenicity, and doses below it, where you don't. So all of this is very suggestive, but actually, can we do some more work to prove that? Well, we explored some metabolomics on this. So we measured the lipid profiles in maternal blood and maternal liver, and also in fetal blood, and we saw a nice dose response of malonyl-CoA, which is the first component after the ACCAs, plus changes in HMG-CoA and succinyl-CoA in the liver. We did dose response in the glycerolipids in blood, which is what we see here, where you see a clear dose response pattern. This is just a PCA analysis of those data, but effectively the non-toxic doses were all clustered together, and the teratogenic developmentally toxic doses were above a certain threshold or degree of lipid perturbation. So we have a picture that says, okay, well, if you have exposure is higher than the potency at the teratogenic doses, altered lipid profiles at the teratogenic doses, the ACCases are embryonically lethal if you knock them out. We've got all this linking information about the relevant biological pathways in the knockouts. And the killer for us, actually, we went back and found another chemotype that binds to a different site on the ACCas, and it produces exactly the same phenotype. So it seems to us that actually we took a long time to work out that we had a mechanism-dependent developmental toxicity. So we then explored these other 66 genes, as I said before, that have developmental signaling and polarity specification defects, which is similar to the kind of profiles you see here, but also these could be other potential molecular initiating events that could cause this. So we haven't formally excluded these other 66 potential molecular initiating events, but I think we've got pretty strong evidence that it's likely to be ACCas in this case. But how does this relate to the developmental tox, the dartable genome effectively? And I think it's trying to generalize what we saw there. So you could hypothesize that if a chemical were to cause developmental toxicity, it interacts with a particular protein sufficiently, so that in vivo you get exposure above some threshold ratio, and that target is, when it's knocked out in mice or mutated in humans, is known to produce a developmental phenotype, and that protein is known to be associated with pathways of development. Those could be your candidates for the dartable genome. And sort of using that kind of thought process, we started curating a list of all of these. Now, historically, we were aware of 58 potential molecular initiating events that could drive this. That actually relates out to 122 genes from the proteins that we started with. But using the principles that I've described on the previous slide, there are some things that we might be dartable genes. So can we identify those using public databases and chemoinformatic approaches? I'll describe that in the next couple of slides. There were some things that we were unaware of, and one was a failed ACCase from one of our legacy companies from about 20 years ago that we were completely unaware of, but we probably should have known. So actually mining the historic data is a key learning actually that we need to do a lot more of, and that's part of what the HESI DARTable genome project is doing. And if we don't know about them and don't need to know about them, perhaps we don't need to worry about them just yet, because actually it's a pretty big problem. So if you ask the question, how big is this dartable genome? Just based on knockout phenotypes with developmental effects of any type in mice, there's quite a large list, and this is the hairball of a lot of those genes and their interrelations as described by Reactome. If you then look at the polypharmacology of some of these targets, how many of those are actually known to interact with known chemicals? If you look at can you measure those in vivo? Well, okay, you've only got a few thousand molecules you can measure in binding assays. Can transcriptomics do better? We're going to explore this question later in the presentation. But actually, do we need to predict everything? So we actually looked at historic studies, and there's only about 150 findings we ever see, and typically 3 to 5 reproducible malformations per class. So perhaps we can sort of prioritize some of the gene lists. So some of these questions are also being explored by the HESI project. So when you put all that together in the numbers, it turns out there are 622 genes that might be in the dartable genome, and a further 3,700 which could be dartable, that we'll need to worry about. But when faced with these numbers in Syngenta, we said, okay, how on earth are we going to build our quantitative knowledge of these AOPs, given the size of this problem and the amount of time and money it had just spent us to to do this on one compound. And that, for us, was when we started having conversations with HESI DARTable, the Developmental Tox Committee, on creating a DARTable genome working group. So what this group is aiming to do is curate our knowledge of molecular initiating events and the adverse outcomes that they trigger. Use this knowledge to see how we could build a DART safety screening profile that didn't rely on the in vivo studies in rats and rabbits. Work out how we would house this information in an updatable public database. So this is a public good for everybody to use. And whilst we're building all of this information, actually explore the process of doing this, identify where the key knowledge gaps are, so we can help people say, actually, if only you made this information available, we could collectively do a lot better at predicting developmental toxicity. So obviously the basic model of the AOP, the quantitative AOP, as I've just described, is the sum target site potency to biological potency ratio. If it goes above a threshold, it triggers various other effects, including some biomarkers, which might be transcriptomic based, that results in toxicity. Okay, this is so far so good. But what actually I showed you was we used a surrogate measure of exposure. Now that kind of worked for one example that I just described, but is it general? So what we really want to do in the DARTable Genome project is explore some of this. So we start with, if you like, a data model and where you might look for that kind of information. So starting with abnormal developmental phenotypes from developmental toxicity studies. They're caused by some teratogenic process. We'll skip over that black box for a second. And I've shown you, hopefully, that can be triggered when a systemic exposure is greater than some threshold. And that threshold is actually what we want to learn from the data. So how do you understand what that threshold is? Well, you have to understand what the systemic dose is, so you need to extract the pharmacokinetic information. But equally, you need to also link that chemical to the protein binding affinities or the phenotypic effects it might have in cell systems that gives you some potency measure of the chemical at a particular target. So you can relate that to the systemic dose to create these thresholds, much the same way as I just described with the ACCAs. So that's the theory, and that's the model we're trying to explore in the DARTable Genome project. So some of the questions we're asking is basically things like, are the affinities and exposure data consistent with a particular protein? And are they consistent between compounds? So I'll come back to that. So current activities. So we only started this project really in December last year. So far, we discussed a number of times our working definition of a quantitative molecular initiating event, and how we would define our data capture templates and our strategy for collecting data. We agreed what the case studies would be, and I'll describe these in more detail in a minute, and their initial goals and deliverables. We've compiled and collected data on 19 ligands so far, and evaluated that data for critical gaps that would stop us doing the data analysis. And we started the analysis on the retinoic acid case study, which I'll just describe next. So what we're doing right now really is just testing the utility and feasibility of this data collection to inform that model using only publicly accessible data for the retinoic acid receptor, AOPs, and also for thalidomide ultimately. The idea here is that once we've done this proof of concept that it can be done using public data, we'll extend it to as diverse an array of compounds as we can that then can be somehow logged in some public database. There's a lot of somehows and some things in this because we haven't worked out how we can do that yet, and that might be an area for fruitful collaboration as well. So where are we? So we're currently identifying, or have identified, as many possible molecular initiating events in the retinoic acid system as we can. We'll get to thalidomide later, and we've identified the relevant PK data. So, oops, I was expecting that to move on. There we go. So we've done a bit of work collecting that data, mostly from the literature and from regulatory submissions. We haven't moved on to modelling yet, and we might need to, but we'll come back to that. We've identified potentially molecular shifting events, mostly from the published scientific literature, but also from downloading from Campbell. We haven't looked at other data sources yet. And we're at this stage right now, which is testing the data that we have collected using this model. So answering these kind of questions around PKs, binding affinities, and threshold ratios. I would just highlight one of the key things that we identified early on, which is actually there's the real paucity of exposure data giving kinetic information for our molecules. There are a couple of papers and a couple of times we can get this out of regulatory documents submitted to FDA, but generally people don't seem to curate their pharmacokinetic information on compounds. And I think if we want to develop quantitative AOPs, this is a key problem we're going to have to deal with. There's loads of toxicology data, it's all over the place, relative, but it's all in unstructured formats, so you have to spend your time extracting it out of papers or out of reviews. The MIE data is, certainly in our first cut, it was mainly focused on occasionally regulatory documents, but mostly it was papers. We've since done a lot more about getting data out of publicly curated databases, in particular Campbell. So we did this for about 19 compounds, and this is effectively the matrix of how good it was, effectively. So to do the analysis we need potency information, some pharmacokinetic, toxicokinetic information, and some tox data. And actually, when you filter what we did, this is only true for 5 compounds, and 2 of those compounds have a different MIE to the other 3. So in reality, we can only do the analysis for 3 compounds, which is somewhat disappointing for something as well studied as the retinoic acid system, but still, there we are. So this is it, really. This is the data. So what I've tried to do here is illustrate what we were trying, the kind of analysis we're trying to do. So if you start with the first compound, alitronitoin, there's at the NOEL, in this column, we've got the ratios of potency of the receptor, RAR alpha receptor, to systemic exposure at the NOEL concentration. The systemic exposure was measured as a steady state concentration derived from the AUC values taken in the DevTox studies. And the same was taken at the low L's, but as you can see, the ratio is a bit higher. So exposure goes up, ratio goes up, and that's a toxic dose. So if you look at this one, this first compound, it's got the highest potency against retinoic acid receptor alpha, NOELs, so up to 1.3 as a ratio can be a NOEL dose, and actually that's consistent for the NOELs of the other compounds. There's nothing higher than that, but the NOEL dose is 2.9. Certainly one of the compounds higher than that and one isn't. But this one at the bottom actually is more potent on retinoic acid receptor gamma than it is on alpha. So this highlights one of our first problems, is the first example we took is actually a difficult one because there's a receptor family. And what do you do about receptor families? But in general, apart from responses in RAR beta, which I'm going to have to explore in a little while because it appears to be an outlier, there's quite a good correlation between the potency ratios that you see at RAR alpha or gamma and the effect and no effect levels. It's consistent between compounds. It's only RAR beta that seems to throw us a bit of a spanner in the works. And, oops, there we go. We've done the same with rabbit as we did with the rat. Only 2 compounds have relevant data. We effectively lost the PK information for one, and one of the compounds doesn't even have a no effect level to explore it against. What you can see is the ratios are much smaller in the rabbit in this case, but again, broadly, it's consistent with the hypothesis. So but whatever the threshold is, it doesn't appear to, at least at the measure of the surrogate exposure measure, it doesn't translate across between rats and rabbits. So we have this interesting sort of like hint of an interesting set of data, but we need more data to test these thresholds. Just the kind of examples we have aren't enough. Another problem we've identified is if you look at the MIE data, the potency data from various sources, they have very different values. And if the inter-assay variability is greater than the difference in exposure between no-L and low-L ratios, and what does that mean for reproducibility and reusability of these data? It's all just lost in the noise. Do we always have to have some standardized protocols to measure these things rather than just taking any assay that's available? One of the biggest problems is the AUC data was not always available. And indeed, often only Cmax concentrations are available and not AUCs at all. So we might need to build on this by modeling out some of these steady state concentrations. There are some outliers, and that might be telling us something useful. It might tell us actually it's all a load of rubbish, of course, but it could tell us that RAR beta isn't actually a trigger for the teratogenic response. Or it could tell us that those particular compounds, they're ones with some metabolism involved. So we haven't accounted for placental transfer and distribution into the fetus, nor have we counted for any effects of half-life. So you might have the same AUC, but actually the Cmax might be much higher, it just clears much faster. So those temporal aspects of the kinetics we might need to explore as well. And so those are ongoing activities within the group right now. So what's next? So we are beginning to address this question of whether other key events could be useful for prediction. Now, at the moment, it must be said this is basically a detailed analysis of the phenotypes used in or observed in the DevTox studies, but it could equally be expanded out into other omics or transcriptomics based reports. So you might imagine, for instance, if you've got a repeat dose toxicology study and some transcriptomics, you might have evidence for the retinoic acid receptor pathway being perturbed in some system. And is that useful for prediction of teratogenic doses? So perhaps you don't need to do the DevTox study, just knowing the pathways perturbed might be enough. Ultimately, we'll move on to the thalidomide case study to move to a different mechanism again. But in the future, we want to begin to collate some of these data to try and identify which in vitro systems might be used to cover all of the identified molecular initiating events. There might be high throughput, high content assays, transcriptomics assays, combination of those with specific binding assays, we don't know yet, but I think there's something there which is definitely an overlap with available data and fits very well, I think, with new approaches. Similarly, we want to build an ongoing process to collate and hold all of this information as with the knowledge develops iteratively, because it's clear with a challenge as large as this one that we won't get the answer straight away, but we need to be able to collate that information through time. And I'm going to finally just point out that actually this is possible to an extent. So we did some work with a compound. You don't need to know what this compound is, but I'll tell you in a minute anyway. And we were just evaluating the L1000 data for does it identify dartable genome? And so this compound in the THB1 cells, actually quite a low concentration, was very highly correlated with mycophenolic acid and very anti-correlated with these inhibitions of proteasomes and the knockout, in this case, of a proteasome component. Now, mycophenolic acid, if you don't know already, inhibits purine biosynthesis. This compound is actually a pharmaceutical DHOD inhibitor. We were using it as a control just to explore this question. It's a teratogen, and we know that the knockout mice for both of these targets are embryonically lethal. So we know that both pyrimidine and purine synthesis give similar responses and anti-correlated with inhibition of proteostasis. So actually, when there is an opportunity to identify signatures of the dartable response, I think systems like this are possible. It's just can we understand that and generate the data well enough to use this in the real world. So clearly, as I said, the L1000 data can provide mechanistic or similarity information around the dartable genome, but only provided there are suitable examples either through chemical treatments or gene knockdowns in the reference database. So there might be, as we start working through this, a need to generate more reference data. Similarly, it's clear that responses are not specific to a particular protein target and molecular initiating event, but perhaps represent cellular responses that are common to groups of MIEs, and that actually might simplify our problem. In that, if we can measure responses at those integration points, it might be much easier. So I'm just going to finish up what I have to say by just pointing out what I think I showed you. We can identify molecular initiating events, so they direct targets of exogenous toxicants. They have a particular profile of systemic exposure versus potency ratios. Typically, they have known developmental biology associated with their function and knockout information that also gives rise to developmental phenotypes. And we can measure abundance changes in transcriptomics. It's just a question of how do you link these two sets of data together to start saying, okay, we're fleshing out these biomarker responses. Now, what I hope I've done in this whistle-stop tour is just showed you that it's possible to identify all of these features, but only if the relevant data is in the public domain. And that, I think, is probably the biggest challenge. So just to finish off with the working group, this isn't the work of myself, it's myself and lots of other people. And thanks to them for their engagement with this project in the last 6 months or so. We have got as far as we have. And I will stop there. Thank you. And we can go through the first set of questions.
Thank you very much, Richard. That was a very elegant and well laid out presentation. It's an extremely hard problem to tackle, as anybody who's done developmental toxicity knows. You have these maternal fetal units, a lot of processes that you can't measure directly. And so it's really an elegant approach that you've taken here to try and identify the potential targets, measure changes in those potential targets, and then relate those to in vivo exposures. I guess my big question is, I think the long-term goal here maybe is to, well, maybe the ACCase model, you have an enzyme inhibition assay, measure Ki, and then you relate that Ki to internal exposures, and whether it reaches the threshold or not predicts dev tox. I wonder, maybe when you identify all the protein targets, you might be able to identify or develop some assays. But in the interim, I wonder if would be easier to maybe use a model like Zebrafish to try and test some of these hypotheses in a system that might be more amenable to measuring molecular endpoints and measuring exposures.
Yeah, and it's funny you say that because we did try Zebrafish, and actually they don't give you a good response for the ACCase class. But that's not generic. I think that's a specific thing that they just appear as really toxic. You don't get a fish being produced effectively because you don't get we think anyway, you don't get closure around the yolk sac and it just lyses. But I agree, actually. Can you explore this in a more amenable system? We've explored actually internally, at least, using C. elegans to do some of that. We've played a little bit with the fish. We've done whole fish transcriptomics, for instance, and you can see mechanism reflected in that. So I think you're right. There are other easier ways to explore some of the molecular details without getting into the mammals. But the challenge comes in when you try and translate that. How do we relate the quantitative measurements of those from one species to another? And that is the real problem.
Yeah. I'll open up to the floor. Are there any other questions that people would like to ask Richard? I see Cyril has written one. So Cyril asks, is Eldert's group, I guess Eldert Pearson's group, exploring this more extensively in Zebrafish, or at least proposing to do so? So what do you know about that, Richard?
So yeah, so Eldert took a proposal to ESITOC to do this in using Zebrafish. So yes, is the short answer to that question. I think the request for proposals based on that is out now. I'm not sure whether it's closed yet, but it will be due to start next year. So yeah, there's an opportunity to do more data generation around that. Exactly.
Mm-hmm. Tish Patel writes, was the ACCase inhibition Ki value for humans or rats/rabbits?
Oh, yeah, I'm sure you can imagine the conversations we had around that. The actual answer is humans, because the assay was available. But yeah, we're still debating whether we should go back and do it within the species. And I think when you see the differences you saw there, I showed there with the retinoics between rats and rabbits, perhaps it is important to do species-specific ones to actually get to the quantitative measure. We don't know that yet, but it's certainly suggestive to me that that's what we might need to do. And we can't just translate one across all mammals.
Yeah. Follow-up question from Atish. What is the organ system, maternal fetal, that can be used to collect and assess the genomic data for DevTox study? I think this gets at an important question of the maternal compartment. Is it predictive of the fetal, or do you actually have to be looking at the fetal?
Yeah. Well, I mean, from a blood collection perspective, you can collect cord blood, but actually, in terms of if you're measuring the fetal response itself, yeah, I mean, you could argue you could take anything, especially if you're playing with the retinoic acid system, because it's got impacts pretty much anywhere. But it's then you wouldn't be taking it from the DevTox study itself. You'd be taking it from a special study with a shorter duration, I would imagine. And I'm not sure that there's a generic one that would work under all circumstances.
Mm-hmm. So a question from Alison Harrell. Thresholds for bioactivity are going to be at least somewhat context dependent. You've looked at species differences. Have you thought about expanding to look across inter-individual variability within a species? So say different strains of rats, for example.
We haven't done that yet. It's true. It is an interesting one, and I must say I do wonder whether we can model inter-individual variability. That's remarkably difficult to say. By the inter-cell variability, if we went to a single cell analysis, I'd quite like someone to explore that question as well. But I think you're right, Alison. There's a lot of things to explore yet.
And Carol Yock asks, I was wondering if transcriptional profiling in embryonic or iPS stem cell models and ability to interact and ability to inhibit their development or cause toxicity as an approach. Could signatures be different for different MIEs and stem cells as a preliminary assessment?
So we did some work with the Stemina DevTox Quick Predict induced pluripotent stem cell model, which I don't know if you're aware of it, but it allows you to, it's a metabolomic based assay that predicts developmental toxicity. But we added on gene expression profiling to that, and you can quite clearly see profiles of molecular initiating events, just the same as you can in other in vitro toxicogenomics based systems. So I think you can probably do a lot more with those kind of assays and get more information out of them. So I think it certainly could be, yes.
And Cyril asks, you noted that one of the challenges is accessing the needed information in the public domain. Is that because it hasn't been released into the public domain, or because there is not sufficient information collected in the privately held databases? And depending upon your answer, what are the opportunities to help surface or gain access to these data?
Yeah, it's a good question. I think it depends on the origin of the chemical. I think more than anything. I think there's a lot of perhaps pharmaceutical and certainly some of the more recent ag chem molecules where there are pharmacokinetic data of relevance that aren't released into the public domain. Perhaps because the regulatory, certainly in ag chem, because the regulatory PK studies aren't actually used for toxicology purposes. They're used for different purposes. Now, and in some cases, the data probably hasn't even been generated. So it's a mixture of some being hidden away and not published versus not being generated. And I think there are opportunities, and the other opportunity I would expand that out so actually and say, we spend a lot of money identifying a particular threshold for these ACCAs examples. I'm sure other companies have done similar things for other mechanisms. So I think you tend to know what you've worked on, but you don't always publish what you've worked on because people view that as a competitive advantage. And can we reframe the way people see toxicology information as a public good and a public service to share it, rather than a competitive advantage in companies? And I think that's one of the biggest challenges we face, is not everybody holds that view, that sharing of the PK and toxicology information to enable society and us all actually to predict toxicology faster is necessarily a good thing.
Mm-hmm.
Yeah, along those lines, do you think I think the EU is coming out with a new mandate to make all toxicology data public? Do you think that will help uncover some of these data?
It might. It might. It depends how broadly that applies. But yes, it is things like that where people either are forced to or voluntarily choose to share the information for a purpose that we can then reuse. Because I think that the key point is that there's a lot of useful information out there, but it's really fragmentary. And how do you get at that? It's just in the set actually that we showed you, we identified 19 molecules that we knew to be retinoic acid receptor agonists because they're listed as such in drug bank, but we can't actually find any data that shows what their potency is against the retinoic acid receptors. Now, it must exist, right? but otherwise they wouldn't be described as retinoic acid receptor agonists in drug bank. But can you find it? That's the challenge.
Right. I have a question. So you know that I think you're aware that the molecular POD group in the HESI DART committee has a model in which we're suggesting that we don't need to know the mechanism to predict a point of departure.
Yeah.
We can just use transcriptional data and bioactivity across the dose response. I wonder if the same couldn't be said for the DevTox, where maybe you don't need to know the MIE or the KI of potential targets. You just need to know where you begin to see molecular change, and would that then predict DevTox?
I think. I mean, I'd like to generate some data to explore that hypothesis actually, because it would be a really good way to not do lots of developmental toxicity studies if you didn't need to. I can't imagine that it won't work out that way, and perhaps the experiment that explores that is Aldert's proposal with the zebrafish. Coming back to your original question, Cam, we explore that in some of those systems and show that there is no phenotype at doses with no transcriptional change for relevant genes, or in fact possibly even for any gene, and that might be enough to allow us to assess the risk of developmental toxicity.
Mhm.
I think that's fine. The problem, of course, comes in those regions of the world where just knowing there's no risk and therefore it's a use is safe is not adequate to achieve registration. So the European Union, basically, where you have to describe the hazard. So predicting the hazard and the doses at which it occurs is very important from that perspective. But I think you're absolutely right from a risk assessment perspective. There might be an opportunity in this to use that data or analogous data in the same way. It might even be the same endpoints, of course. That's something to explore as well, actually, as you develop that data set. Is it protective of all risks?
Yeah, certainly if you know the MIE, you pin that down, then you can get at the human relevance question and get at more of the hazard question, I believe.
Yeah.
Question from Jean Yao. Your assessment on using linked L1000 data for toxicology purpose question.
Yeah.
Are the compounds annotated with safety info?
No, not really. There are some papers linking it to, linking some of those chemicals through to known adverse effects that have occurred in the clinic, but I haven't really seen a lot linking it to toxicology information in preclinical studies. And the data that we had, we'd done the toxicology on our own molecules before we did the L1000 data sets.
So, question for me, you mentioned potential collaborative opportunities between the DARTable Genome Working Group project and the HESI DART committee.
Yeah.
What's kind of your vision for that?
Well, I think it kind of builds on the conversation we're just having around, can you use gene expression markers as a key event effectively? So can we replace or use biomarkers of effect as opposed to the apical endpoints, and those being measured at the level of transcriptomics? That's one. The other is actually when you consider the point of departure project you're working on, does that number always also protect you against the potential for developmental effects? That's just to, it might require a different categorization of where your data is, but you'd know that, right? If somebody's analyzed whether the DevTox effects, if they exist, are always at doses higher than the ADI, if you're predicting the ADI conservatively, then you're already protective. So it's just to explore that question as well. And I guess the third area of collaboration would be around this, the kind of in vitro molecular initiating event identification part of the work. Can you use transcriptomic data or high content data of any sort really to help build these systems models of the dartable pathways? So I showed, purine and pyrimidine inhibitors activate the same pathway. To what extent, if we try this with other molecules, would we start getting a coalescence around commonalities of pathways, etc.? So I think it's using that kind of deeper knowledge of that data that might help us with the identify MIEs, but also identify biomarkers for the dartable effects.
Yeah, happy to explore more of that with you. Question from Philippe Coutet, I think so I pronounce the name. At which stage of embryonic development do your compounds become toxic?
Oh, we don't know. Is a short answer, and depends between compounds, I would imagine, because we've not actually done the window dosing studies to test that. Given that we've seen quite a lot of with the ACCAs, certainly the body wall closure defects, it's probably quite late on post organogenesis, and it's around growth. Pulling the GI tract back in and folding the body around it. So it's probably later stages really for that one. But equally, we've got other chemistry which affects organogenesis, and they have very early effects, sometimes on the head, sometimes just on general growth. So it's not a question that's just a simple answer.
Yeah. And follow-up to that question is, which organ is most affected typically?
Do you mean in terms of in adults or during development?
Yeah, I believe the question is maybe what organ is the most common teratogenic organ?
Oh, right. So it's usually the head in terms of what we've seen recently. But it's not always, as I've showed you. We saw defects in the tail and defects in body wall closure.
I guess a related question to that. Often, fetal body weight change is an extremely common, at least in my experience, developmental effect, not necessarily teratogenicity, frank malformations. Do you think that this project could also address fetal body weight changes?
Yeah, we've debated that. It might, but that's going to be quite that takes you into quite a different or challenging area, effectively, of biology, because you've got a lot of fetal and maternal communication and interaction to deconvolute to work that one out. So whether it's and I think that would be useful, because, as you say, it's a very common finding, right? But we were trying to focus on, okay, can we get this to work on a well-known, well-established, and clear teratogenic mechanism first? I think it could be expanded, though, ultimately.
Any other questions from the group? I don't see any. So on behalf of the E-STAR committee, I want to really thank Richard for his very nice presentation today. Richard, if you would like to follow up with the committee, we'd be more than happy to continue the discussion.
Excellent. Thank you. Thank you for listening, everyone.
Thanks so much, Richard.
Have a good day, everybody.
Thanks all.
Thank you.
All right, thank you.
Bye-bye.